everybody, welcome to the stream. It's been a little while, and I'm very excited to get to talk to you all about basically the subject that I've been teaching up until this very point. Biological engineering. What does that mean? How do you do it? And what stories do I have from the field? That is what we're going to be getting into today. But before that, I'm going to say hello to everybody. Hello, Alexandros. Hello, Velvet. Hello, Artistic. And N-methyltransferase. Awesome name. Hello, Someone and Celeste. Welcome. Hello, Katie. Hello, 44 CPU. Welcome. Hello, Dreamless. Welcome. CDM. Welcome. Human Buddy. Hello. Oh, the chat shifted. Hello, CR Smith. Welcome. Of course, Toxo. Welcome. Hello, Mylan. Welcome. Hello, Mylan. Mylan, my fellow V-Theorem member, psychologist. Hello, Otto. Welcome. More Juice. Welcome. Lunkel. Welcome. You can just call me Seed. Will do. All right. Let's already get started. So to be very clear, to be very transparent, this is an introduction to biological engineering. If you want to talk about things like replacing Drosophila antennae with legs, that will be for a separate stream. Today, I'm only going to be covering the groundwork for how to think about engineering in a biological way, and I'm going to be sharing some of my favorite stories from this field, but not all of my favorite stories, because then why would you come back and watch me later, right? So today, I'm only going to be sharing 3 specific stories, but I hope that they will be enough to hook you for when I talk about some more exciting things down the line. Very exciting stream today. Will you ever make a video on Plasmodium falciparum? So that is the malaria parasite, and the answer to that is very much yes. A video about malaria is something that I have been thinking about since I basically founded my channel. It's just there are other things to do before that. So absolutely, Plasmodium falciparum is definitely coming. Definitely. You can just call me Seed, call me Panzer. All right. Very good. Alrighty. Did you see my message on Discord in the Victories channel? I did not. Take a quick look at that. Beat my executive dysfunction today. That's awesome. All right. Very cool. Let's get started. Why don't we? All right. Let's get into it. So what is biological engineering? It's become kind of a meme about biological engineering that it's very hard to actually explain this to anybody. So one of the common complaints that the students had in the major that I was teaching in is that they had no real idea how to tell their parents or their family members what it is that they do. And I think today we are actually going to be enabling those students to be able to share with their parents, their families, etc., what it is a biological engineer actually does. Not at all, Astral. You are just in time here. We are just getting started. So what is actual biological engineering? And, there are many definitions. Here is mine. Biological engineering uses and or interfaces with biology to solve problems. Very broad, but I word it in this way because I think of engineering as a discipline that is involved in solving problems. So this is going to be in contrast to basic science, which I will contrast this with later on in the stream. But this is to get you started to have an idea of what constitutes as biological engineering here. All right, so as some of you in chat have already predicted, we are going to start off at the very beginning of biological engineering, back when biological engineering was just agriculture, right? So this is in principle biological engineering's great grandparent, and what agriculturalists have been doing since the dawn of mankind is not in form that different than what a biological engineer does today. So we have a problem. That problem is hungry. And you have some starting material. This is the Brassica Acacia. I don't really know how to say it, but this is a wild plant. And through many, many iterations of domestication, you come up with a solution. You notice that, hey, nibbling on this plant, there are a couple of edible parts that aren't going to poison us or kill us. And these two parts are the tasty leaves and the tasty buds. And you start noticing over the course of domestication of this plant that if you pair the plants with the tastier leaves with other plants with tastier leaves, their children will have tastier leaves. You do the same thing for the buds, and eventually what the effect is you get an actual differentiation of this wild ancestor into something actually edible. So these are all technically the same species, but they are different cultivars. So while they are, the same, again, like exactly like the same species, they look very different, they taste very different, but that is because we have put in many, 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 many years of careful cultivation to select for traits that we desire. So this fits in with this format of having a problem, engineering a solution, and having an effect, right? So that kind of idea, that kind of flow or structure is, I think, how you should think about not only biological engineering, but engineering in general, right? And this is just one out of like a bazillion answers to this, but I wanted to show you that what biological engineers do is not that different from this. Now, the approach might be different, but it's always taking something from biology and fixing something. Hey, Clockwork, 5 week indeed. But we're actually not going to be talking about a guided evolution, although that will come up in a video that I'm not about to put out, but then my next video, which should hopefully be out in about a month, is going to go over some of those ideas. So stay tuned for that. So the birth of molecular biology in the 1950s actually accelerates the field of biological engineering. So before molecular biology in the 1950s, biological engineering consisted of like making medical devices, but we didn't really have the molecular tools to actually be able to engineer in the way that we do now. And so the simultaneous advancements of the fields of molecular biology, chemistry, physics, medicine, mechanics, mechanical engineering, electrical engineering, all of that combined to form this very young discipline of biological engineering. So it's a pretty new field relative to other fields, of course. All right. While much of the biological engineering that took place in the mid-20th century focused on medical devices, for example, the MRI, for example, was invented in the 1970s, the advent of molecular biology meant that we could have a much broader definition of what biological engineering could cover, which is pretty interesting, right? So not only can we think about biological engineering on the microscopic level, but it can go up to a macroscopic level. MRI is that old? I know, I was pretty surprised too, honestly. So we can go from the microscopic, right? Proteins, DNA, RNA, pharmaceuticals, biomolecules, to a little bit bigger in the form of microbes, cells, tissues, animals, plants, and then finally even whole biomes and ecosystems. So biological engineering spans across so many different size ranges that it's difficult to cover all of this in one stream. That's why I'm only picking 3 of my favorite stories, trying to pull from a couple of different levels here. All right. Here's a note that I want to make clear for my audience, just so that if I'm unable to answer your questions, you understand why. I was never trained as an engineer. I was, of course, trained as a basic science. Basic science will always be my first and foremost love in this discipline. But I think that because I came from basic science, I have a pretty unique perspective on how engineering works, which I think is pretty neat here. So a basic science education requires that you read deeply. Of course, you need to know all of the lore in your field. And so I really recommend basic science if you are okay with that, right? Because when I talk to engineers, they don't really like reading as much, which is totally fine, honestly. However, engineering and engineering education requires that you play and solve problems. And also, when you do read in engineering, it's so that you can accumulate tools in your toolkit. And I think that is an important distinction. So there are, of course, overlaps, right? Scientists and engineers will have, I don't know, like 80% the same classes in undergraduate. It's those 20% left over that defines whether or not you become a scientist or an engineer. Although, again, those categories are not mutually exclusive, right? So if you're taking a lot of math classes, learning how to do things, that's, leaning engineering. And if you're just learning stuff, that's leaning basic science. Yeah. More juice. You're going to make me blush. I'm not going to read that out loud. Me sitting and waiting for CRISPR. CRISPR is coming up. CRISPR is, I think, one of the best stories ever told in biological engineering. Yeah. So in terms of teaching, I'm not going to be able to do as much in terms of teaching you how to engineer because, of course, I'm not an engineer, I'm a scientist. But in terms of teaching you the basics, it's probably more important that you understand the engineering mindset. And so in order to do that, let's look at a couple of different case studies for creative inspiration. I think that's probably the biggest difference between me as an undergraduate thinking that engineering wasn't for me, is because as an undergraduate, as a freshman, I never thought I was smart enough, weirdly enough, to be an engineer because I did not have confidence in my ability to solve problems. I think in order for me to have been comfortable solving problems, I would have needed first a basic science education, because when you get marketed as a freshman for the different fields, it's like, yeah, problem solving. But when you're 18, there are not that many problems that you have the tools to solve. And so I was always scared off by the field of engineering because I didn't have the raw intelligence, I think, to be able to problem solve in that way, and also not the background. So I think it's pretty interesting in that regard. Not to say that engineers are smarter than scientists, it's just that I had a misconception. In your engineering education, you will learn a toolkit, and then after you figure out your toolkit, then you can go off and start solving problems. I would recommend that you not be as afraid as I was back as a freshman to take on engineering as a discipline. You will learn how to solve problems as you go. All right, let's now continue here. So here are the different case studies that I have decided to talk about today. And it's mostly going to be focused on this first one, but I added these two because I think they touch upon some nice applications of engineering, as well as different levels and different problems with regards to engineering. And of course, as I've teased, not only in the thumbnail, but also in this talk itself, we are going to start off with CRISPR-Cas9, and also a technology that spun off of CRISPR called CRISPR-I. And of course, if you are a biological engineer, please do not spoil the story. When I ask questions, I am primarily going to be asking them to people who are unfamiliar with this technology. So because I think that it is important as a storyteller that we remain in suspense. That's to say, if we can't squeeze out some answers from the non-scientists, non-engineers in the audience, I will ask for your input then. But for now, as we continue on in the story, please allow the suspense to build here. I think that is fair. All right, so to tease the rest of the stories, or I guess I'll just quickly define all the stories so you get an idea of what we're talking about. CRISPR-Cas9 is, of course, the revolutionary technology / discovery that really brought forth a new era in biological engineering, because it made editing an organism's genome almost trivially easy. I say almost trivially easy. There's still some constraints to CRISPR. CRISPR's not perfect, but it made it so that you could do it very cheaply, and you could teach high schoolers to do it at the benchtop without without much specialty equipment. It really made the process of being able to work with more biological tools that much easier. Golden Rice is a story about engineering an actual plant, so engineering an actual organism in order to solve a very severe metabolic deficiency or nutritional deficiency that a lot of the world actually has. And so this is going to be a nice story to show you how we can leverage engineering biology to help a lot of people who are in need. And finally, we have the small molecule microarray. This is perhaps a technology that is not as well known for non-scientists and non-engineers, but of course, if you are a biological engineer, you are probably at least passingly familiar with this technology, and I wanted to point this out because it's a really neat instance of how you can have a very basic idea that anybody could really come up with and have it solve a huge problem and really contribute a lot to the field of drug discovery. So Artistic asks, or Panzer asks, what's a microarray? We will get to it. We will get to it. All in good time. Alrighty. So, of course, we are now going to begin talking about CRISPR, not a CRISPR, but rather CRISPR, a revolutionized, a technology that revolutionized biological engineering, and we are going to talk about the story behind this. So even if you have heard of CRISPR, even if you know how it works, I want to talk about CRISPR as a scientist because I think the story is often something that gets left out of the educational material. Sources for this? You caught me, surprise pants. I didn't include my source for there being a difference here. All right. So this is the first case study. What is CRISPR and how does it work? We are going to talk about the history behind this discovery because I think it's honestly really creepy. So we know now that CRISPR is a technology that biologists can use to precisely edit the genome of an organism. So, you know, every organism has a genome, a bunch of collections of DNA inside your cells. That DNA codes for mRNA, which then gets converted into protein, and that protein is largely defines how your biology functions, right? So to be able to precisely edit the blueprint of life is, of course, you can imagine, pretty huge here. All right. So CRISPR, maybe to the surprise of some of you, was not invented by humans. CRISPR is actually the natural immune system of bacteria. I actually have a video on this. It's one of my first videos. It's about the war between bacteria and phage. So phage are viruses that only infect bacteria. And so, yeah, humans did not make CRISPR. We took CRISPR from bacteria and adapted it for our purposes. Common prokaryote W, exactly. What does CRISPR stand for? We will get into that. So, but I just want to appreciate right now that the DNA editing technology that we are now using like crazy came from the humble bacteria. So what is CRISPR? CRISPR stands for clustered, regularly interspaced, short palindromic repeats. This is a mouthful, but we will go over exactly what this means. And so in order to do that, let's first begin this story, right? So in 1987, researchers in Japan accidentally discovered a region of the E. coli genome that was honestly really spooky. There were repetitive sequences of DNA that were separated by interruption. So this is a very simplified cartoon here, right? Don't take this as an actual CRISPR array. That is just, you know, a way for me to illustrate. So in red here, we have these repeats, right? GAATTC, GAATTC. So there are repeats here and they are separated by unknown, or rather poorly defined chunks of DNA. And it was unknown for a long time what this thing actually does. And I just want to take some time to appreciate how actually insane this finding is. So for context, these researchers were trying to sequence the gene for alkaline phosphatase isoenzyme. So a completely unrelated metabolic enzyme in the bacteria. The researchers were like, hey, we want to know the DNA sequence that codes for this protein. And then they get back their sequencing data and they find these spooky repeats. You know, these repeats, GAATTC, again, not the actual repeat, but an example. And it happens periodically. And they're spaced, right? So this is clustered. They're all in a line, regularly interspaced, right? These repetitions followed by this spacer happen very frequently, repeating one after another. These repeats are interspaced with some other DNA that we don't really know the function of, and they are short and palindromic, and we will discuss why they are palindromic. They don't look palindromic like you would expect. So for those of you who don't know or might not be familiar with this word, a palindrome is a word that is read the same backwards and forwards. So for example, what's a good palindrome? Noon, right? You know, noon as in afternoon, Noon, you read it the same back and front, noon from the front, noon from the back, right? Yeah. So They don't look like palindromes, but we will see ways in which they can be described as palindromic. All right, so very, very freaky, very, very freaky here. So yeah, this is really weird and totally unexpected, and pretty much a discovery by accident, which happens quite a lot in the field of science. So I kind of imagine these repeats as like ancient aliens, conspiracy, like very weird, bizarre, creepy energy that is hidden in the genome, hidden in the code of the E. coli, right? Because it's not entirely clear if this would make a protein and what protein it would make. All right. So before I explain what the CRISPR stuff actually is, we first have to remind ourselves what the rules of base pairing are. So you know that you have DNA. This DNA is the code for life. It codes for mRNA, which then is converted into protein. But you have two rungs of the ladder of your DNA, right? DNA is a double helix. It has two rungs and they are bound to each other. So this is, of course, a cartoon of the helix unwound, untwisted, I should say, not unwound, but untwisted. So we have two strands of your DNA, top strand and bottom strand, and there is a directionality of these strands. So in order for scientists to be able to describe which strand they're talking about, they order them based on the chemical structure of the DNA. So you don't really have to know why these are called 5' and 3' ends, but they are. So if we read the DNA sequence from left to right here, this top strand is 5' to 3', and then this bottom strand is the reverse order. So this rung runs in the exact opposite direction. So when we read now from right to left, we are reading from 5' to 3'. So this allows us to orient ourselves with regards to which side of the ladder that we are actually on. And we have to know that in terms of DNA, that there are set base pairing rules. So guanine always pairs with cytosine and adenine always pairs with thymine. If you are studying for the MCAT, you can very easily remember this as GCAT, GCAT, GCAT. That's how I remember it, at least. Right? Yeah. So that is that. Yalda, you should start Patreon. I actually have YouTube membership, which I prefer, even though Patreon takes less of a cut than YouTube membership. I think it's a lot more convenient to just, to support me through membership because it's already in the app. And I can also take advantage of the YouTube platform to actually give my members something unique in a way where it is much more complicated and difficult to do for Patreon or Ko-Fi. So if you actually click !membership, you can be reminded that I have new membership rules, which is something that I was going to talk a little bit about later, but I have updated my membership incentives. So you can go ahead and check those out if you try and join my membership, which I think, the button should be in the description here. All right. So, wait, no, they don't. I thought bacteria only had RNA. So yeah, bacteria actually are DNA based. Viruses, as Lincoln said, can either be DNA or RNA based. So yeah, so to remind you all, we have these base pairs. We have these base pairing rules. A's always pair with T's. G's always paired with C's. So what makes these repeat sequences so weird, right? So these repeat sequences, these GAATTCs, again, not the actual repeats, but it is weird that these regularly repeating sequences are all palindromic. They read the same from back to front when considering their complement. So again, I said we're reading from 5' to 3'. This sequence is GAATC, but when you read DNA strand 2 from 5' to 3', look at that. You actually see the same sequence, right? GAATTC, reading from left to right. And then when you reverse that direction, you actually end up reading the same word. So that is what that is. Yeah, so I want to pause here and kind of poll the chat because this is a really interesting point that is going to be really important for understanding why this is so freaky. So I'm going to start a poll here. Do you understand why GAATTC is actually palindromic? And I'll leave this up here while I take a sip of water. Yeah, chat, do you see the palindrome, right? Do you see the palindrome? These repeating sequences, they're really, really freaky. It's one thing if they were just repeats, but they are repeat palindromes. GAATTC from 5' to 3', and their complementary strand is also GAATTC, reading from 5' to 3'. I get it, but it's weird. It is weird, isn't it? It's very, very weird. So I'll explain it one more time. Why is this freaky? No, I do not at all. If DNA only binds to one kind, shouldn't it always be palindromic? So you can generate a random sort of DNA sequence and test it for yourself. They're not always read the same back to front. I get it, but why would this keep existing? Yeah, it's weird, isn't it? Why would these palindromes continue to show up? And oh, by the way, CR, I also had that thought, and I had to like do a sanity check. Yeah, if you test it out by making a random, do like TTCCCCG or something, and try that. They are complementary to one another, but they're not always read exactly the same from back to front. Why would a bacteria waste its DNA on this? Yeah, it's a good question. It's a good question. It's really freaky. It's very weird. Very freaky, very weird. So what is this information doing? And now I hope that you are starting to see why I find this to be like ancient aliens, like conspiracy, like what is going on here? Because you have these palindromes and they keep on repeating and they are regularly spaced, and who knows if they make protein. I mean, they probably could attempt to tell if they make protein, but if they make protein, what does this protein do? Why would it have these weird repeats that are separated by these sequences? Very, very strange. That's my strange emoji. Yeah. So repetition itself is not that strange. Actually, repetitions happen a lot in biology and in the genetic code, actually. So the thing about repeating DNA is that this is something that I'll get into a lot in my next video, but the thing about repetitions is that repetitions in DNA can happen because DNA polymerase can just kind of slip. DNA polymerase can just slip. It can accidentally peel off from your DNA, and in repetitious sequences, you can actually have an accident where DNA polymerase copies over the same strand of DNA and adds another exact copy at the end. This happens not infrequently. Well, it does happen infrequently. It doesn't happen infrequently and then cause something good to happen, is what I'll say. So if you want to know more about how DNA polymerase skips and why that's important and why that's cool, keep an eye out for my next video about E. coli for more details. But yeah, this idea of repeating DNA, that's not the weird part to me, because repeating DNA happens all the time. DNA polymerase is kind of messy. Do you mean skip or slip? Sorry, that should be slip and not skip. Thank you so much, someone. It should be slipping. But these CRISPR repeats are, of course, spaced for some reason. So if it's the regular DNA polymerase slip, these repetitions should be back-to-back roughly. But the reason why this is extra freaky is that these repeats are regularly spaced for some reason, and it's very, very bizarre. So what is the function of these palindromic repeats? Why do these freaky sequences of DNA keep showing up in the bacteria? And the thing is, I'm not going to say. In terms of building the suspense here, these little freaky sequences, I don't think should be explained at this part of the story. I will explain what these repeat palindromic sequences do in the future, but hold on to this idea for now, because I want the mystery to build. So we are not going to take a look at these palindromes right now, but keep this in the back of your mind as being really weird to experience these repeating palindromes. So instead of looking at the palindromes, we're going to look at the other sequences. So just to recap a little bit, you're here, you're a Japanese scientist in the 80s, you're trying to sequence the gene for this particular protein here. You're trying to sequence this particular protein here. And by accident, you come across a whole region of mysterious repeating DNA polymerase palindromes separated by cryptic codes. This is bonkers, very, very strange, and it's not at all obvious why you have those repeating palindromes. But before we talk about the palindromes, let's first talk about the spacers. So 18 years later, the identity of the spacers was revealed. And now I will ask the audience what they think these spacers are. Not biologists or engineers, or if you know the answer, please keep it to yourself. I will take some time to have the non-scientists, non-engineers, or people very early in their education to guess. We know that this sequence, this weird region of the DNA, is somehow responsible for bacteria killing phage. Bacteria killing phage. So, what is, or what do you think these little tiny bits of DNA in between the palindromes could be? We know that this is a sequence that enables bacteria to kill phage. And I want to hear what my non-scientist, non-engineer audience thinks about this. Leftover viral replication genes? Treb, I'm going to need you to elaborate on what you mean by that. But does anybody else have a hypothesis? Leftovers from viruses? Interesting. Correction and errors? Maybe it's a section that allows recognition of phage. Okay. A proto-immune system? Okay. Interesting. Like operons? Okay. Let's get a couple more guesses and then we will see who amongst the audience is right. And I know that my scientists and engineers are probably screaming the answer, but that's, I'm not asking you. If you know the answer, that's totally fine. But this is just for people to have the space to imagine the answer, because I think that's more fun. Antibody synthesis. So I'm going to correct you on that, human. Bacteria don't make antibodies. That's a eukaryotic thing. But something maybe akin to antibodies, maybe. Striking back the enemies with their own weapons. Certainly an interesting hypothesis here. Okay. So now that we have had a little bit of time to think about what these repeat sequences could be, let's do the drum roll. What are these sequences? Where do they come from? They come from phage. These pieces of DNA that are in between these mysterious palindromic sequences come from bacteriophage. These are pieces of DNA that the bacteria has chewed up and memorized in order to power the machinery of their immune defense. So now we can finally put together the story. We have regularly interspersed palindromic repeats that are separated by pieces of DNA that come from viruses that kill bacteria. And again, it took 18 years to go from the Japanese scientist accidentally finding this mysterious ancient aliens-esque part of the bacteria, and it took 18 years to find out that the stuff that's separating these palindromes actually come from viruses. Very, very, very crazy. Love this. All right. So in 2007, definitive proof was published that CRISPR is a bacterial immune system. And so this is the title of the paper. You can already guess it's paywalled, so I can't show you this paper. But if you are an aspiring biologist and you've taken Intro to Biology, either in high school or in undergrad, I highly recommend that you actually challenge yourself to read this paper through your university, because again, it is paywalled, very annoyingly so. And I actually remember being forced to read this paper when I was a student as well. Not when it came out. I'm not quite that old. But this was required reading for me, and it really shows you in brilliant detail how the scientists figured out that this sequence of DNA is responsible for anti-phage defense. Sci-Hub? Yeah, could be. Alrighty. So, how does the CRISPR phage defense works? So this is a simplified cartoon that I got from Wikipedia. And so I will kind of walk through. Yeah, I think a lot of you were kind of right, Katie and someone. And that goes to show that there's some level of intuition that happens over the course of natural selection that you can kind of suss out. Okay, so how does the bacterial defense system work against phage DNA? So phage, you have here in the upper corner, the phage is going to insert their DNA, and this DNA is going to be made into copies of this phage and then kill the bacteria, which the bacteria do not want. So the bacteria have evolved a way to destroy phage DNA. So if you're a biologist, you know that this happens in the form of restriction enzymes that chew up viral RNA or DNA. But the bacteria are actually more clever than that. Bacteria can actually memorize the phage DNA so that they can destroy them much quicker next time they encounter the phage, kind of like our own systems of adaptive immunity, which I think is incredible. So you squeeze in that phage DNA and you have some enzymes, Cas1 and Cas2, that cut a piece of that DNA. And through a bunch of biochemistry, you can actually incorporate that tiny slice of that phage DNA and insert it into the bacterial chromosome here, right? So I'm not going to go into detail how the protospacer is acquired. Not enough time in the stream for that, but that is how that initial acquisition happens, so that you can establish a primitive adaptive immunity in bacteria. This is the simplified version. Oh, it gets much more complicated. But in order to enjoy the CRISPR science, you don't need to know that detail, honestly. Yeah, artistic and dreamless. Yeah, it does remind me of our own immune system. Except for us, it's a little different, but it does evoke the same feeling of like the bacteria can memorize. It has a memory of all the phages that once infected it, which I think is really, really interesting. So this happens when the bacteria survives the phage. Yeah. This is if the bacteria gets infected by phage and they survive, they can pass off this memory when they replicate, so they can gain phage resistance through CRISPR. Are there regular palindromic sequences kind of like making the DNA harmless? That's an interesting hypothesis. I don't know if anybody can realistically correctly hypothesize what the palindromes are for. It would take a really brilliant insight that I wouldn't have to do so, but it would be fun to think about it. And I'm happy and I'm glad that you are hypothesizing, despite the fact that I think it's so out of left field that it would be challenging to come up with it without a biology education. As Kazakhs would say, consume and adapt. Yeah, exactly. Exactly. It's not just a feature of eukaryotes. Bacteria can do that exact same thing too. You can study immunology for your entire life and still not know half. Exactly. I totally agree. Totally agree. Okay, so once you have that immune memory, how does this bacterial immune system actually work? So you have transcription of the sequences, and part of the CRISPR system is an enzyme called Cas9, which makes this enzyme. This enzyme is an enzyme that can cut viral DNA. So this part codes for this enzyme, and then the mystery sequences then code for pieces of DNA that bind to Cas9 and help Cas9 identify future viral infection and cut their DNA. So this is like a restriction enzyme. However, it's a restriction enzyme that can adapt to any phage that tries to attack the bacteria. Restriction enzymes are kind of pre-programmed to recognize certain sites, which is not very adaptable, right? This CRISPR system is a true acquisition of immunity, where the Cas9 enzyme teams up with the memory of phage infection in order to detect that sequence and cut it up. So, does that kind of make sense, Chad? I want to pause here as well on this screen to get a sense for whether or not you get the gist of how CRISPR works, while I take any questions in the meantime. Yeah, so cutting up the viral DNA, stuffing it into your own genome, into your own bacterial chromosome, and then using that memory alongside the Cas9 enzyme to cut up that specific piece of DNA next time it shows up. Is it the palindrome, the gene that codes for the Cas1 and Cas2 proteins? Good guess, but unfortunately no, Treb. The palindromes do not code for Cas1 and Cas2. I learned all this in my biotech class. Yeah, biotech. It would be bad to have a biotech class where you don't learn this. Oh, and Robert, if you came late, then I just wanted to say that like most of the questions that I'm asking to my non-science, non-engineering viewers, just to have the childlike wonder of hypothesizing what the answers are here. What is this poll for? It's just to get a sense for whether or not you get the gist of how bacteria memorize and fight phage infection. I don't get how they are able to insert it in without accidentally disturbing nearby genes. So this space in the bacterial chromosome is just for the immune memory. They have a specialized area in the chromosome where the CRISPR spacers are kept. Now, exactly how it is that they managed to keep that specificity, I actually don't know that answer. It's an excellent question to look up. I don't have that answer off the top of my memory, but if I had to guess, there's probably some sequence specificity with the Cas1, Cas2 enzymes that are local to these spacers. Won't the plasmid bloat? It's a really interesting question whether or not there's an upper limit. I'm sure there's an upper limit for how many of these sequences you can stuff in. However, through the process of recombination, I'm hypothesizing that through recombination you can actually accidentally loop out some of these old memories. So there's again, I'm not 100% sure, but I think you can remove some of these by accident. There's like an equilibrium of things accidentally leaving and then new memories being acquired. I don't know what the frequency of that quote unquote forgetting would be, but I think that it's possible to happen in the CRISPR array. So it's like killer T cells and antigen presenting in one. Kind of, in a way. I think you can draw that analogy, but instead of being protein-based, it's DNA-based, right? When it detects the phage DNA, it starts freaking out. All right, so 82%, that I will say is good enough for now. For those of you who maybe don't quite, for those of you where this did not completely register, I'll try to simplify it and just say that in order for the bacteria to fight the phage, they steal a piece of their DNA so they can recognize it in the future. And when they recognize it in the future, they can cut it up before that DNA has the chance to be made into other viruses. Okay, so let's continue from here. So the engineers in the chat might have some ideas, but essentially what this bacterial immune system is an enzyme that can be guided to a specific place on DNA and cut at that exact location. So CRISPR-Cas9 is very cool, very neat for scientists, but when you tell an engineer about this, they are salivating because now you have a weapon, not a weapon, you shouldn't call it a weapon. You have a tool. It's a weapon against phage. That's why I said that. But for human hands, this is a tool that can help you identify and locate a specific piece on DNA and cut at it. Almost any location you can think of. Almost. It's not perfect, but you can program the system to attack any sequence of DNA, almost, that you could want. And I think that is interesting. Oh, it's a location beacon. Yeah, I think you can put it that way. All right. So now we can talk about these mysterious palindromes. We've identified the fact that these spacers, oops. Oh, no, that's a spoiler. Okay, well, hopefully none of you actually saw that. Hopefully none of you actually saw that. So let's try to build a mystery. Hopefully I tad back soon enough, but we know that these spacers are chunks of phage DNA. These help the bacteria acquire a phage memory. So now we can actually answer or think about what these spacers are for. So now I want to farm ideas from the chat about what these spacers are from. And I actually think that not the spacers, the palindromes. I actually think that it's pretty easy to forget what these palindromes are and why they work. I know certainly I had to remind myself what these palindromes actually do, but I do want to take some time now to sit with this mystery. We know what this piece does. Now what does this piece do? And again, I'm not expecting that anybody actually gets this correctly because it is very, very specific, but there's something fun about trying to come up with an idea even if you're wrong. So what do we think these palindromes are for? What are these palindromes for? I'm gonna delete Robert's comment. No offense, Robert. I want to keep this spoiler free. Maybe the palindrome is something to code for the protein cutting. Not totally incorrect. Something around that. Do not read flags. That's a very interesting hypothesis. I'll ask you what you mean by do not read. A way of signaling the location of memorized DNA. Also a very interesting idea. Like maybe that's a homing beacon for the correct whatevers that need to be localized to that particular area. Also a very interesting idea. An attachment point. Okay. Attachment point of what? Can we be a little more specific? Could they be targets? Targets for what? Splice site or an origin? Splice site, that's interesting. If these are spliced out, the splicing is involved, kind of, not in the way that eukaryotes do it, but yeah. DNA cutting or DNA polymerase, something code for the protein cutting. Yeah, could be. All right. Cas9, and it's not Cas9, I can tell you that, just because Cas9 is located before any of these genes. Viral gene here markers. Interesting ideas. All of you have great ideas. I'm actually not entirely sure how many of your ideas actually end up being correct. I know one of the functions of these palindromes, so if any of the experienced biologists can help fill in the blank as to whether or not chat has had ideas that happened upon other correct answers, I'm very free to having that information filled in. So I will reveal one of the functions of these palindromes, just one. So palindromic sequences can fold in funny ways. You can see that there are two strands of DNA here, and we have a potential palindrome in GATC. And again, we can check that it's a palindrome, right? GATC, that's how that reads in this direction, and when we read in that direction, we also read GATC. So this is a palindrome, and we know that palindromes like binding to each other, and of course, if you have two strands of DNA, it doesn't really matter, that's not that interesting, but palindromes can help singular strands of DNA bind. So look at this structure. This is called a hairpin loop, or I guess this part is called a hairpin loop, because it looks like a hairpin. And we can actually see, if we trace through here, if we follow here, this is one strand of DNA binding to itself, right? If we're looking down here, we're going up, we're looping up and around, and like that. We have one DNA not binding to a partner DNA, but binding to itself in a structure called a hairpin loop. And that's kind of curious. That is curious, right? This is a unique property of palindromes because they are palindromic, because they pair with each other and read the same back and front, that you can have one strand of DNA binding internally, not in the double helix, a two-rung twisted ladder formation, but a local pocket that can form a little bubble, which is very, very interesting. And why is that interesting? It can give this piece of RNA a very unique shape. The CRISPR system actually needs a piece of RNA that fulfills a very specific shape in the Cas9 enzyme called tracer RNA to work. This is why I don't think anybody could have guessed this correctly, and it was just kind of fun to be left in the mystery a little bit. I think all of you had really interesting hypotheses, and I think those authors of that 2007 paper probably tested some of what you were suggesting. So I'm not saying your ideas were bad. You didn't have the full story to be able to put this together. But I am curious, because those are very good hypotheses to test to make sure that those are not the answers instead of this. And again, any experienced biologist can now fill in the blank here. You can feel free to tell me whether or not chat had the right answers before this reveal so that we can get the full story. But I just wanted to talk about tracer RNA. So this is the Cas9 enzyme, and it binds to the DNA, and it can make cuts in the DNA, double-stranded DNA. And in order to do so, it needs a couple of components. It needs the PAM sequence, which is a sequence universally as NGG on the on one strand of the DNA. You don't have to worry too much about the PAM sequence right now, but it also needs the CRISPR RNA. So this is the RNA that contains the viral memory, right? This is the RNA that binds the actual viral DNA. But it also needs, in order to form the correct shape of the enzyme, this piece of DNA called this piece of RNA called the tracer RNA. This RNA folds in a particular shape, and you can see that there are hairpins here. It needs these hairpins to be able to smush the Cas9 enzyme into the correct shape so that we get actual cutting. If you don't have this tracer RNA here, you will not have a functional Cas9 enzyme. So for those of you in chat who said, is this required for this Cas9 enzyme to actually cut the DNA? The answer to that is yes, it is, because it needs to be shaped into a hairpin to force the Cas9 enzyme into the correct shape to do the actual cutting. And I think that's kind of neat. Does that scrunched up bit contain the virus DNA like a vacuole? Not like a vacuole, like an enzyme, like a polymerase does. It forms a tight protein complex, whereas a vacuole is more like an empty space. So this is the tracer RNA. And so what Robert was actually teasing earlier is that when humans have this technology, there's no reason to have a two-component system where you have tracer RNA and the CRISPR RNA. You can combine them in one big RNA called the single guide RNA or sgRNA. So that is what Robert was referring to before. So this is a bit more of a nuanced point, and it's kind of hard. It's not entirely intuitive, right? But to say it one more time, in order for the CRISPR-Cas9 system to work, in order for this Cas9 enzyme to actually do the cutting, it needs to be in the right shape. And in order to be the right shape, it needs a tracer RNA here that has these hairpin loops in order to have the correct shape. And if all the pieces are assembled, this Cas9 enzyme can then cut the DNA and kill the phage. All right. What's the rRNA again? That's CRISPR RNA. This is the strand of RNA that contains the immune memory of the phage in the normal bacterial system. Do our cells do similar things when it notices wrong DNA folds? I know our body does a lot when trying to keep our DNA from imploding. Yeah, so if you get kinks in your DNA, if you have DNA damage where the DNA causes the DNA to stick out in weird ways, the DNA polymerase can actually run over. Well, actually, I don't know if it's DNA polymerase that fixes bumps on the DNA strand. There's an enzyme. I don't know if it's DNA polymerase or something else, but there are enzymes that can fix bumps along the DNA ladder. If it sticks out in a funky shape, you can get that detection and you can fix it afterwards. And what does tracer DNA stand for? It's just to make this part of the system that forces this to be the correct shape. I like how versatile nucleotides are. So many ways to make a knot with the same group of molecules. Exactly. There are RNA nerds out there that love making things out of RNA, and I think that's very neat. You're a neutrophil, shouldn't you know this? You're a human, shouldn't you know everything having to do with being a human? I think that's gonna be my go-to response. For that question from now on. I mean, I don't take any offense to it. I do think it's genuinely very funny. All right. So what the heck are these palindromes? These palindromes are structural features enabling tracer RNA to be made in order to slot into Cas9 for an operational enzyme. This, I think, is a bit harder of a mystery to solve in terms of like making this very clear to people, but I think I will be happy if the audience at least just understands that the palindromes are there so that the Cas9 enzyme works correctly. I think if you get that much from this presentation, I will be happy with that, because it is a little more complicated to wrap your head around. So, to recap, the CRISPR-Cas9 system is a system that bacteria evolved in order to fight phage, and this involves cutting out little bits and pieces of viral DNA, memorizing them by stuffing them into the bacterial chromosome, and then using the Cas9 enzyme to use that transcript in order to identify that place on the viral genome and then destroy that piece of DNA. Okay, so again, this is probably red meat for engineers. Like, this system allows you to recruit an enzyme that does cutting to anywhere you can imagine, asterisk. It's not exactly everywhere, but it's everywhere enough for most people. And so whether or not it's worth engineering something or using this CRISPR system kind of depends on the current state of gene editing. So before we had CRISPR, we had two different systems that could make edits. We had TALEN and we had zinc finger. Now, I am not an expert in either one of these technologies. But before I get into that, someone has a good question. Wait, how does the bacteria not succumb the first time? That's an excellent question, someone. The battle between phage and bacteria is pretty complicated. I do recommend watching my video about it. It is mostly fixed on CRISPR, but I think there are probably other things in that video. I don't remember. It's been a while since I made that video, so I don't remember if I explained restriction enzymes. But bacteria have kind of hard-coded enzymes that destroy things that typically belong to phage. If CRISPR is the adaptive immune system, restriction enzymes are the innate immune system for bacteria. And so whether or not a bacteria survives infection is kind of not clear. And in textbooks and stuff, the phage always win. You never see the bacteria winning except in the context of CRISPR. But in actuality, it's not that straightforward. Not every infection by phage results in a dead bacteria because bacteria have immune systems. It's a good question, though. So yeah, so getting back to this, is it worth engineering CRISPR into something that humans can use? What was the state of genetic engineering before CRISPR? We had talon and we had zinc fingers. I'm not experts in these technologies, but we should inspect some of their basic pros and cons to determine whether or not CRISPR is an exciting way into the future. So before that, let us go over how CRISPR can actually make edits to DNA. So we already saw that CRISPR is very good at destroying DNA. You can guide it using a guide RNA to a specific spot on DNA, and you can have it cut there. And that's very useful, of course, if you need to kill phage. But how can we actually leverage this technology to make edits to the genome? And so here you have the CRISPR system. You have this cleavage. You break the DNA. You guide your system to this DNA. But how do you actually edit, right? How do you have that engineering power? And when you break the DNA, you have two distinct repair pathways. Either the bacteria will do something called non-homologous end joining, or NHEJ, which is their preferred repair pathway. It's more common, but it's a little messier, but it is also faster. And this non-homologous end joining, NHEJ, often introduces deletions or insertions. It's like kind of crappy at its job. It's good enough that the bacteria can survive, but there is a decent chance that you do get a insertion, which is going to lead to a broken protein, or a deletion, which will also result in a broken protein. Remember that if you put in anything or if you remove anything from the genome, you can cause what is called a frame shift, where you screw up how the code is read, right? Code of the DNA is read in triplicates, right? So every 3 bases of the DNA codes for one protein. And if you put in just one sequence in there, you can cause everything to be screwed up because you've shifted the reading frame, which you can hear more about in my video about the flu. A lot of interesting reading frame biology in that flu video, which if you haven't watched, right now, you should probably go do that. It's a great video. I love seeing a figure and instantly recognizing which paper it came from because I've also used it before. This is a great image, very great image. Yeah. Sorry for the late answer, but I was in a Helldivers 2 mission. I don't know what any other uses of the palindromic DNA, but I'm only a bachelor's, not a PhD. That's fair. I think the palindromic thing is mostly for Cas9. Wait, if 3 nucleotides code for protein, then how many proteins are there? Aren't like piles of DNA. So if you read the DNA, right, let me do it this way. Every 3 pieces of DNA, every 3 codes, like AAGG, GGG, CCC, TTT, whatever, every triplet codes for 1 amino acid, not protein. I shouldn't have said protein. So like, I don't have these memorized off the top of my head, but you can imagine that TTT, that might code for serine. I'm fairly certain it doesn't, but let's just imagine in this case. So serine is an amino acid. It's 1 block of protein. So I misspoke. It should have been amino acid. Yeah. So, again, the bacteria, they would prefer to do non-homologous end joining, where they kind of fix the DNA, they kind of stitch the 2 pieces together. This often results in a tiny little bit of DNA being inserted or deleted, which is not ideal, can often break the protein of interest. So if your goal is to just break a gene, you don't really care how you break it, this is a way you could do it. Now, bacteria have a much fancier way to repair DNA called homologous recombination or homology-directed repair. That's this pathway. And so here, this requires there to be donor DNA to fix what is broken. So it cleaves, right? You still get those two pieces, piece 1, piece 2. You still get those two pieces of DNA, but if you give them donor DNA, the bacteria can actually use the donor DNA as a kind of template to remember how to fix what is in this gap, right? So you break the gap, you introduce an additional piece of DNA that contains donor DNA, and that donor DNA has to be flanked. Like the bacteria have to essentially think that this donor DNA is the correct template, that this fixes the DNA. And in order to do that, you need to make sure that you flank your donor DNA with the same sequence that is surrounding this particular portion of the DNA. And when that happens, you get a process called homology-directed repair, where you trick the bacteria into incorporating your donor DNA. Super, super cool here. Again, this is a bit more advanced, and if you haven't had as much background in biology, this is going to be kind of hard to grasp. So I will go over this one more time. Most of the time, bacteria just do this. If you don't give them a donor DNA, they just break the DNA and then they kind of repair it kind of crappy. And a lot of the time, you can get the crappy repair breaking the gene that you're trying to edit. And if you just care about breaking a protein, that's totally cool. Where would one get donor DNA from a setting outside of a lab? You kind of don't. The donor DNA is something that you make and that you put into the cell. And so here, we have a very high potential pathway for engineering. If you introduce donor DNA, you program this DNA, you just print this DNA out, and you print it such that you flank your interesting sequence of DNA in the middle with the gene, or with the DNA that comes from the bacteria. You can get a situation where the bacteria get tricked into thinking that this donor DNA is a healthy, normal template. So they will take the template and they will repair the broken DNA based off of that template and get an insertion of a sequence. Pretty incredible. The old switcheroo, exactly. It is a switcheroo in that regard. How big of a block can it cut? It's not so much cutting, because you can do the homology. I think you can do the homology and also have it be... Actually, I don't remember. That's an interesting question. So this is in the context of insertion, but I actually want to see how this can be adapted for deletion if it's recombination. Like if you wanted to delete a specific segment of a protein. I still think it's through homology-directed repair. I just don't remember how deleting a chunk works. Is this their DNA? The donor DNA in the context of engineering is DNA that you introduce as the researcher. You put in that DNA. How does the donor DNA get to its specific site in the genome? Through, you put enough of the donor DNA where it floats around to the point where statistically it will find its cognate pair, right? So this piece, again, that's part of tricking the bacteria into thinking this is the healthy template, is that this DNA will eventually kind of bind to this region just because it has that homology. So because they have the same sequences, they can base pair with each other. In fact, you see that homology in here, right? So if you have this floating piece of donor DNA, the two strands will kind of unwind and bind to each other because they're so similar. That's kind of the basis for homology here. This looks like a bit like recombination during meiosis. Is it related? It is absolutely related. Same sort of principle involved in recombination. Looping out of the DNA for deletion. Yeah, I don't remember exactly mechanistically how that works, although it is possible here. At least I thought that looping out was for deletion, what I can come up with on the spot. I see. So not incredibly confident in that answer. It sounded good to me. I'm not entirely confident I could reproduce from memory what the deletion pathway looks like. But just know that it is possible. Pure chance, Brownian motion. Yeah, so Brownian motion, pure chance, whatever you want to call it. Brownian motion will get the DNA to the right spot, and then homology allows it to actually do the recombination. Good biochemical principles to keep in mind, or rather thermodynamic principles to keep in mind. Okay, so I will take a poll here. Do you get the gist? Maybe not 100% because this is, of course, kind of sticky, crunchy molecular biology going on, but do you get like the gist of what is happening here? You can essentially repair, you can trick the bacteria into repairing their broken DNA in exactly the way that you want it to because you can give them donor DNA. I think that is what I would like for you to get out of here. If you understand the principle that breaking the DNA and then repairing them in a different way allows you to edit the DNA, that's what I want you to get out of here. So if you ask the question, how would you edit the DNA? Just like as a vague conceptual question, one way you can do it is you break the DNA and then you repair it the way you want it to repair it. Why is there a fork in the DNA in that middle section? So this is what is known as homologous recombination. This is a feature of DNA where because the sequences are so similar, they can get kind of tangled up, I think is the best way to simplify that. Homologous recombination will be the bane of any genetics student. So that is a sneak preview if you are going to be learning a lot about DNA in your formal studies. I say that, but it's actually really cool the way it works. It's just kind of complicated when you first start learning how all of this stuff happens. Why is there a fork in the DNA in the middle of the section? If you're referring to these two right here, that is because bacteria have two repair pathways, either non-homologous end joining or homology-directed repair. So they have two strategies to repair their DNA. But how do you break the DNA? So the CRISPR-Cas9 enzyme can do it, right? So the CRISPR-Cas9, that's this enzyme, you gave it a guide RNA that tells it to be recruited here. That guide RNA also contains that tracer RNA that enables the Cas9 enzyme to slice and dice. So with all of those components, you get that breakage, and that breakage is then repaired. If you don't introduce donor DNA, it'll be non-homologous end joining, where it's kind of crappy and kind of whatever. If you give it donor DNA, you can repair the DNA in exactly the same way you want it to be repaired. How large of a block can be inserted? I think actually there was a paper recently where a huge chunk of a gene was inserted. I didn't actually read the paper in time because I was changing slides up until the hour before the stream. I originally was not going to do any of this. I was originally going to like breeze through CRISPR, but I figured it'd be kind of fun to talk about the mechanics of CRISPR. But I think recently the size limitation of CRISPR has been improved quite drastically. So I can't give you an answer, but it's pretty big from what I understand. Okay. So 90% of you get the gist of it. That's awesome. That's a great ratio. You can post the link in the Discord, Lunkel. For those of you who haven't joined the Discord, !Discord for that. Sorry for the multiple questions, but how does the RNA thing actually cut the DNA? Like chemically, what does it do? I understand. Oh, okay, so that is kind of an on-the-spot question from me, and my background in chemistry is kind of weak. So I do not remember the exact order of how electrons move through. I'm sorry, I'm kind of unprepared to show you how that works. However, it is mediated by chemical reactions. And if you are the type of person who likes chemistry, we can certainly find resources for you to look at the diagrams of electrons moving and bonds breaking, but I'm unable to do that for you right now. Okay. So why is this so tempting for engineers? What makes CRISPR so good compared to Talon or zinc finger? I'm not going to talk about zinc finger. I'll just use Talon because I find that I think Talon is the more recent, more high-tech version of the zinc finger stuff. Talon is another gene editing technique. It's not useless, it's not irrelevant because CRISPR exists, but it's certainly not appealing for a large set of problems to solve. So Talon is protein DNA based, meaning each target needs a new protein to be engineered. So this is, of course, annoying, costly, and expensive, whereas CRISPR is RNA DNA based, meaning each target just needs a new guide RNA to be made, which can be printed for cheap, right? If I were to buy a piece of RNA, you could do it. I don't remember how much you pay for a single guide RNA. It's not, you could do it pretty like normally. I think it's like engineering a protein, that's expensive. That's thousands of dollars. Buying some RNA is pretty cheap. I'm not going to say the number because I don't want you to actually do this, right? This is not a how-to CRISPR. this is how CRISPR works stream. But the barrier to this is that almost every molecular biology lab is capable of doing a CRISPR experiment, whereas doing a Talon experiment requires more specialty stuff, which is not accessible. You can pay people to print RNA? Absolutely. Actually, what you would actually do is you would pay people to print DNA. You would use the DNA to insert into E. coli, which can make RNA for you very reliably. And then you would ask the E. coli to do all the hard work for you. Actually, you would ask E. coli to make the entire CRISPR thing. I'm not going to get into the details of that because, again, this is not a how-to CRISPR stream, just a what is CRISPR stream. But yeah, printing DNA is like arbitrary. And that is, of course, not without its risks, right? You should not just be printing random sequences of DNA. But if you are a researcher who understands the risks and how to mitigate those risks, you can certainly do it for very cheap. Yeah. I'm just very curious as to what is actually happening at the lowest level. We can look at before it gets overwhelming. I say before the quantum level. Yeah, it'll be at the chemistry level, which I'm not that good at. A bunch of electrons and bonds being broken. I just hand wave it as organic chemistry. Maybe you could do a how to CRISPR stream. I don't think I should because I do not want to empower everybody to do so. You should only know how to do CRISPR if you are in a situation where a human watches over you and makes sure that you are not doing something shady, right? Releasing an organism out into the wild, not disposing of your GMOs properly, doing something that is dangerous otherwise with foreign DNA. How to CRISPR would not be something that I would do for the public. It's just not, I don't think it's very ethical to do so. But the principles behind CRISPR, I think, is very ethical to share because it's interesting, and also it's important to know how these things work if you are interested in biology. I know a group that still works with Talons because they can recognize epigenetic modifications. Yeah, I think most of the genetic editing problems can be done with CRISPR for cheaper, but Talon is irreplaceable because it has niche use cases, which I think is very interesting. Now you're going to tell me I shouldn't use CRISPR to remake the variola virus? I mean, I'm saying that's in the realm of things that I want to prevent people from being able to do. Not that you would be able to do it anyway. Like, actually putting this all together is like pretty hard, but I don't want to be involved in that happening. Like, my responsibility ends. Like, if you're interested in CRISPR, you want to learn more about CRISPR, that's on you, but I'm not going to put the power in your hands, right? That's on you, not on me. Okay. You can kind of imagine the rest from here, though. Like, CRISPR is so much cheaper and so much more effective than Talon. And that is why CRISPR is such a big freaking deal. I mean, now it's not that sexy to be talking about CRISPR in the field. Like, the undergraduates nowadays are already kind of bored of CRISPR, which I find to be incredible because when CRISPR was like around when I was studying, it was like the hot thing. And now undergrads are bored when you mention CRISPR. It's just a given to them, which is kind of insane. Like, you do not know how hard it was to do precise genome editing before CRISPR, and you are taking it for granted. But yes, genome editing with CRISPR has exploded, enabling easy manipulation of an organism's genome for all sorts of application, from basic research, right? Like, if you want to break a protein in a very specific way instead of just generally, like, you could break a protein by putting another gene into it. In fact, if you watched my prion video, you would know that in research studying what the prion protein did, the classic way to break the prion protein was just to put another entire gene inside of the gene for the prion protein, right? That's just what you would do to break proteins. You just put a random antibiotic resistance gene inside of an existing gene, and that would pretty reliably break that protein, but it was taking a hammer to a problem that doesn't need a hammer. Now you can tweeze the protein, right? You can see, okay, I suspect that this specific arginine is important for this enzyme's function. Now you can just CRISPR it. You can just say, okay, we are going to replace that arginine with a serine and see if it works, right? So it's been very important for basic research. Metabolic engineering, right? If you want to engineer a bacteria to make a commercially important product, say ethanol, right? If you want to do something in the biofuels industry where you want to manufacture ethanol, you can use CRISPR to maybe screw around with the way that E. coli does metabolism to get more ethanol out of it. So that is a possible application. Biomanufacturing, so that's along the same lines as metabolic engineering, where say you want to engineer spider silk, right? I don't really know what goes into spider silk, so this is a random example that hasn't been proofread, but say you want to make a lot of spider silk, you want to engineer that. Now you can do so more easily with these precise editing techniques. And of course, controversially, gene therapy. So of course, gene therapy, editing the egg and sperm, editing the embryo, that's like very, very no good in terms of ethics. We shouldn't do that, right? And so engineering can never be separate from ethics and morality and philosophy, because most of us kind of agree that you should not be making super babies. However, gene therapy with regards to changing the genome of the cells that are dysfunctional, I think that most people are pretty happy with. Say you have a liver disease because your liver is not making a particular enzyme correctly. Well, you can now do gene therapy where you can imagine a gene therapy where you edit just the liver cells, so not the eggs or sperm, but just the liver cells, to now begin making the protein as you would need it. So that is an important nuance to think about with regards to gene therapy. Oh my, maybe we won't even need to kill silkworms in a horrible way anymore. Yeah, exactly. Like if you can leverage precise gene editing, I mean, you probably might not even need CRISPR, honestly. There are other methods, if you, depending on how tricky it is to make spider silk or silk in general, that might not require CRISPR, but you can certainly imagine CRISPR could be very useful with regards to solving this particular problem. Okay, but CRISPR isn't done yet. Modifications of the Cas9 enzyme allows for engineers to do all sorts of crazy things. So here's a separate problem. What if you didn't actually want to remove or knock out a gene? What if you merely wanted to reduce the protein production? How could you modify the CRISPR-Cas9 system to do so? And I'm going to give chat a little bit of time to just ponder this engineering problem, because I think it's an interesting problem. And unlike the previous question that I asked, this one is solvable by people who do not have engineering or biology background. So why would you want to knock down instead of knock out? Well, if you are working, say, with E. coli and you want to futz with their metabolism and you want to decrease the amount of enzyme but not completely remove it, because if you completely remove the enzyme, the E. coli dies, right? So just completely deleting a gene, that is a very strong way to control the E. coli's metabolism, but you don't always want to be that strong. You don't always want to go that hard. Sometimes you want to just be a little more gentle with your approach. How would you modify the system to do that? Produce less Cas9? Not quite where I would go with that, just because that would just result in fewer bacteria getting your engineering product. Modify the Cas9 to add epigenetic tags to the target sequence. I mean, that sounds awesome, and that would probably work. I could see a way that would work. I wouldn't actually be surprised, Alexandros, if your answer is out there somewhere, but that is actually not the answer that I am looking for. Could you insert DNA to break DNA in only certain parts? If you break it only in certain parts, you still break the protein, and the protein is what's important here, Day. So if you break the protein as a whole, you just prevent it from working entirely. You can only edit one allele depending on the gene. So a sided CRISPR. So I think that CRISPR can do only one strand. I think it's been a while since I've read CRISPR papers, but yeah. One allele. Yeah. So bacteria, they're haploid. Yeah. I see what you mean. Yeah, I thought you were talking about which side of the DNA wrong. Modify the promoter to have less bonding affinity. Actually, that technically is correct. Like you could use CRISPR to modify the promoter to do that. That is actually true. That's an excellent strategy that kind of circumvents the answer here. So gold, you do get a gold star because your answer does work and technically does answer the question, but I'm thinking more about how you modify the CRISPR-Cas9 system itself. Because you have it so like Cas9 knocks out some of the amino acids so the protein isn't as efficient. That would require a lot of information about how the protein works, and oftentimes in the field that's just not practical. So it's not a bad idea, it's just an idea that would be in practice pretty hard to do reliably. Yes, there are variants that only cut one strand. Thank you. I thought I had remembered that. Okay, so this is, I think, a really brilliant way to do things, where you could just break the Cas9. This is a super interesting and great engineering approach because it is so simple, right? So the goal is to adapt this DNA cutter to instead of cutting DNA, just prevent the protein from being made as highly. So instead of, and again, Chad, you had a lot of great answers that are technically correct. It's just not the correct answer I was looking for. So don't be offended or anything, or don't think your answer was any less wrong. But in terms of the answer that I was fishing for, you could break the dCas9 here. You could break the dCas9 to make it so that the dCas9, so that the Cas9 can't cut DNA. So dCas9 is deficient Cas9. This is now an enzyme that does everything that Cas9 does except cut DNA. So what does it actually do instead? Instead of cutting DNA, you can route this deficient Cas9 to this exact spot on the DNA, and then the enzyme just sits there. Then the enzyme just sits there. And if the enzyme just sits there, then that portion of the DNA can never be read to make RNA. And if it can't be read to make RNA, then it can't be read to make protein. Is that some sort of artificial repressor? It is kind of like an artificial repressor, although repressor has a very specific... Oh, no, you're right, Mr. Winner. You're right. A repressor, yeah, you could use the word repressor. I was going to be very finicky about the definition of repressor, but I think it's fine to use the word repressor. Smart. Yeah, exactly, Seed. Like, it's very smart. And that's why I think engineering is so fun, is because you go... Like basic science is very interesting because you can marvel at how clever nature is. Engineering is very interesting because you can marvel at how smart humans are, right? So this is not a perfect 100%... This bacteria never makes the protein again because, of course, as I've said before on videos, biology is just jiggling shapes bumping into one another. So a non-0% of the time, this Cas9 enzyme pops off of the DNA and a little bit of protein can get made. But most of the time, this dCas9 enzyme will sit on that DNA and prevent the production of mRNA, prevent the production of protein. And again, I love this as an engineering idea because it is so simple that it makes you wish you thought of it. I also can't take full credit for some of these examples. I learned a lot about this on the job, right? So I can't take credit for coming up with some of these examples because I kind of inherited a lot of these examples. Obviously, I would give credit, but giving credit would dox me, so I'm not going to do that. But I did want to take some time to acknowledge that these ideas are not all of my own. So with the proper background, CRISPR-Cas9 serves as an incredible engineering sandbox for creative students to play in. But of course not without thinking about the ethics/societal impacts, of course, right? Like we have to be very diligent, and even though engineering can be very fun, like it's very fun to make things, it's very fun to blow things up and, just work, it's fun to play Minecraft. What is Minecraft if not engineering, right? But if you want to do this professionally, if you want to do this seriously, you have to always be thinking, am I making a product that is going to be bad for humanity? And, this is especially hot in AI engineering right now, right? And it's always hot in biology, but you can see with your own two eyes many ways in which engineering has made life better and many ways in which engineering has made life worse. So just be aware of that. And I don't really, we already talked a little bit about the fact that using CRISPR-Cas9 to edit germline cells, that is, a lot of people agree, not cool. But as long as you're using it in an ethical way, it's incredible. Okay, case number 2. There should be some kind of environmental application for it. Oh, for sure. Oh, yeah. Like if you are trying to, for example, fix, what is it called? Photosynthesis, Protein complex, gosh, what is it called? Chat, please. Chat, I know you know what I'm thinking of. The protein complex, inefficient, carbon fixation, Rubisco. Thank you so much, Alexandros. If you are trying to solve climate change by trying to engineer a better plant that can suck up more carbon dioxide from the environment, CRISPR is probably one of the tools that you would consider for that strategy, to engineer and design a better Rubisco. I don't know if it's the smartest tool, but you can certainly imagine you could not be blamed for trying it, I would say. I'm sure that if a state actor wanted to, they could use this unethically. I mean, that is why we have to be very diligent about making sure that this technology only is used for good, essentially. This is why STEM can never be separated from the humanities, right? If you are a STEM student and you crap on humanities students because blah, 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 your thing is so easy, blah, 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 you make no money, which happened all the time when I was an undergraduate. Piss off? Is that a little spicy? That's like my biggest pet peeve about stuck up STEM people, is that they do not give credit to the people in the humanities that are keeping society from consuming itself. So again, take a bunch of humanities classes if you are taking STEM classes as well. Don't just focus on STEM, or there's a non-zero chance that you are going to lead to the downfall of humanity. All right. I tried to look at the Cas9 mechanism, and I think it has two ions acting like a base to stabilize the transition state, leading to DNA backbone getting broken up. Certainly the case. That is a macro level. I was more thinking about like the organic chemistry level of all the bonds moving around, but I think that's a pretty good explanation for it, Mr. Winner. That's probably an explanation that if I had in the back of my head, I probably would have said, so thank you. Did they use CRISPR to make those sterile male mosquito populations to help control malaria, dengue? I don't know if that was a product of CRISPR. I feel like when I heard about that mosquito, it was pretty early, so I'm not actually sure. That is rough. I'm afraid that it could outcompete native populations. It's possible, which is why maybe the smart thing to do would be to grow them in some kind of contained greenhouse type thing where you can't get cross-pollination outside of it. But it's also like this is very real, right? Like there is no engineering without risk, right? Every time you make something, there is going to be risk. Now those risks can be quite devastating for biology. But you know what else is devastating is doing nothing while our planet burns and floods. So that is why I am an advocate for biological engineering with regards to climate change, even though I do acknowledge that there are risks, and there should be no implementation of a technology that has not been rigorously analyzed for safety and containment, right? Like this is something I treat very seriously, but I also want to provide a bit of that nuance where there is a risk to saying that there is a risk to everything. It has to be a calculated risk, right? Like that is what is underneath all engineering, is calculated risk. My streamer is spicy today. I'm a little spicy today, I think. As someone who is a humanities major, I agree. Yes, I very much agree. It's so annoying to hear that. It annoyed the hell out of me as a STEM student myself. I heard they were already doing it here in Brazil. I did read that they implemented some of this gene drive technology somewhere. I don't remember the details of it though. I hear the guy who invented leaded gasoline also invented CFCs, even without ethics. It can't be worse than him. Fair enough. Yeah. Can you sterilize the plants in the second or third generation? This is something that I think is really interesting that I will cover later, is engineering of GMOs for climate change. I don't want to spoil their strategies, but there are technologies out there that address your concern about sterilization, not in the way that you think it does, but in a way that I think is cool. So I will save that as a spoiler for a more focused bioengineering stream, right? Because today is just 101. Today is just, you hear me yap and hopefully you get interested in studying some of this for yourself. And then I will come to that specific engineering trope case study in the future. So keep your eyes peeled for that. All right. So let's talk about golden rice finally, right? So golden rice, I've been on this slide for a long time. So golden rice, time to actually talk about it, is a GMO. I know that people don't like saying that, but you are my audience. You are a biology inclined audience. And so I will be honest, it is a GMO. It is a genetically modified organism. It is a rice that was engineered to solve a very severe problem. So vitamin A deficiency is a pretty nasty condition worldwide, leading to increased infection and blindness. You can see how much vitamin A deficiency actually affects so much of our actual globe, right? In terms of determining the percent of the population that actually lives in these areas, it's a pretty huge chunk, right? And so if you have vitamin A deficiency, it can lead to increased infection because vitamin A deficiency is immunosuppressive and can also cause blindness. So this is a huge deal for a lot of the world. And so common sources of vitamin A are from animal products and plant sources. But from plant sources, they tend not to be very bioavailable, although carrots are still pretty good, right? So carrots are a great way to get vitamin A. It's why we associate vitamin A with great eyesight. It's more like carrots prevent you from going blind instead of carrots give you supervision, but whatever. But the problem is that you can't get access to fresh carrots. A lot of plants are not great sources for vitamin A because they're not easily convertible into vitamin A. And of course, animal products, it would do the globe good to reduce our reliance on animal products. I myself am not like a vegan, but I have made a conscious effort to eat meat as little as I can and make the attempt at more ethical consumption. Of course, I'm just one person. What does the relative contribution of populations have over corporations? We can have that argument, but the idea here is, as a society, as a population, as companies, as producers of things, reducing our reliance on animal products, definitely not a bad idea. It's also very expensive for a lot of places in the world to have access to animals that you can eat. So ideally, what we can do is engineer a crop that is cheap, high calorie, able to nourish the people who eat this and help solve this vitamin A deficiency problem. All right. So golden rice circumvents this problem by making the vitamin A precursor, which is beta carotene. So you can very easily tell which of these rices are the golden rice. Of course, golden rice kind of gives it away. It is the actually, it is colored, right? It is golden in color because it makes beta carotene, which is the thing that carrots make. And so this is a precursor that you can then convert into vitamin A. And so how does this happen? We have a little bit of metabolic engineering. So here is the pathway, the metabolic pathway that has been inserted into the rice genome. So you take a gene from the daffodil, this phytotein synthase, right? This gene, which converts this precursor inside the rice to this product, phytoene plus 2 pyrophosphate. And then in order to get beta carotene, you have to take another gene from soil bacteria, CRT1, which then converts this into lycopene. And then once you get lycopene cyclase converts lycopene to beta carotene, right? So this is metabolic engineering. So this is one of those scope issues. So CRISPR is engineering on a genetic level, molecular level. Now we are seeing applications of gene editing or transformation, GMOs. We have a way in which we can stuff an entire metabolic pathway in the form of these two genes, one from daffodil, one from bacteria, into the rice. And again, to make sure that people come away from assessing risk, we are taking a gene from the bacteria. Could that gene elicit an immune reaction? The chance is vanishingly low. This is not an immunogenic protein from that bacteria, right? So bacteria make all sorts of stuff that your immune system does not like, and your immune system will let you know if that protein is around because you will get sick. This is one of those proteins that does not really produce a significant immune reaction. Also, it ends up in your gut, which, your acids will take care of this protein before it ever potentially even becomes an issue. So to, make it very clear, this is an example where you can take something from bacteria and have it be pretty harmless, okay? So if you want to do good for the world and you like biology, this is the kind of thing that you could do, right? Like, we all have different skill sets. And I thought for a long time that the way that I could do good was to become a medical doctor. And during my education, I realized I would be a terrible medical doctor, right? But what I am good at is yapping. And so, just like how my skill set is yapping to all of you and making videos and talking about the subject that I love so much, some of you are equipped to be medical doctors where you can handle that kind of stressful lifestyle and handle that kind of training. And others of you will be better engineers, people who can actually solve these problems. And if you want to do a lot of good for the world, one of the ways that you can contribute is through something like golden rice, right? Like, I don't know that much about the story of golden rice as a whole, but you can see the potential for golden rice, right? A product that is more nutritious and can solve a lot of the vitamin deficiencies that the world has. So I think that golden rice is a pretty inspirational problem where, if you are like me, like, I idolized doctors, I idolized basic research, and I didn't really care about my immediate impact on the world when I was studying. But if you have it in you to want to make a big difference, something like this I think would be pretty cool, I think. Why does rice just happen to have that precursor? What would we do if it didn't? So, Mr. Winter, is an excellent question that came up during the engineering of golden rice. So the researchers that made golden rice actually did think to input lycopene cyclase. They didn't actually realize that rice made lycopene cyclase. So when they were doing the stepwise engineering, they found that, oh, we put in these two genes and it was already making golden rice, which was awesome, right? They made an additional extra discovery. Hey, rice also has lycopene cyclase, which is kind of fun. Very, very neat. Okay. So I love this idea. And if you want to do good by many people, consider biological engineering. Of course, it's not the only way. Again, I just made the whole spiel about each one of us has their own skill set. But if you like biology and you like engineering, you could think about it. Okay, so golden rice is an example of biological engineering that could potentially help a significant chunk of the world that we live in to help them live healthier lives. But of course, we are talking about ethics here. Its existence is not to the liking of everybody. There are a lot of interest groups that are not happy with GMOs as an idea. And to say it very briefly, GMOs are also not without risk, right? We talked about contamination, we talked about cross-contamination, outcompeting natural populations of these things. If there is a selective advantage of this, is that going to wreck ecosystems? But again, there is no risk without, there is no reward without that risk. And so if you can engineer a system in which you can grow rice in a very contained, isolated environment, that might be worth that potential risk if it means being able to cure blindness in so much of the world's population, right? So it's messy, it's complicated, it should be talked about. Whether or not the trade-off is worth it, that is up for debate. And I'm not saying there's a right answer here, but I personally lean on the side of being pro golden rice. Now, where I agree that GMOs have a problem is the use of pesticides, right? So there is there currently is and have been a lot of pushback onto GMO as a concept. But in my experience as a biologist, as long as the proper testing is done, a lot of GMO food is pretty safe to eat. However, in order to grow a lot of that food, you need to use a specific type of pesticide, and that could be pretty bad for the environment. It could be bad for the people that work on these crops. That's another complicated conversation to have. If you want to tackle that problem, you might think about trying to do GMOs in a way that is not as reliant on pesticides, which I think is interesting. Yeah, Alexandros, you got it. Okay. So, case 3. So that is, yeah, and Gabrielle, you got more of that nuance in, and it's a good conversation to have, one that I'm not completely equipped to talk about in this very moment, just because I need to do more reading to make sure that I don't do a bad job, because it is such an important topic. But I will now move on to case 3. But before I do that, Patrikios just had a bowel movement, which I will take care of now. I will be right back. You may enjoy my be right back screen. It should not take that long. Be right back with case 3. So see you all in just a little bit. gosh. Oh, there I am. Oh, oh my God. Oh, I did not even be right back. Okay. All right. Okay. Let's do this. Let's try to get that PowerPoint back. And that is this here. Great. We're doing it. Okay, yeah. So that's certainly the first time that's ever happened to me. Patrikios pooping so hard, my computer crashed, but it does look like we are back now. Hello to all of you. We are talking about biological engineering. Now let's get back to where we were at. We talked about CRISPR, the history behind CRISPR, the story behind CRISPR, as well as the applications of CRISPR, some of the ethics and morality of CRISPR. And we talked a bit about golden rice, a GMO product that is going to help. I think it's already been deployed in a couple of countries, but like it is a rice that has been engineered to produce a lot more, or to help you produce a lot more vitamin A so that you can have good eyeballs. Not blind eyeballs. All right. And now we are in the last third of the presentation. Welcome back, Fi. Did you remember to drink water? I did not remember to drink water. Let me do so right now. Okay. Very cool. Okay. Very frazzled. But we trudge on. Let us talk about the third biological engineering story to get you an idea of what else can be done with biological engineering. And this one's a shout out for all of you chemistry people, because this is more in your realm. So the case 3 that we have here is the small molecule microarray. This is a very neat piece of technology, and we will see what problem it solves. So here's the problem. Mother Nature, as well as chemical labs, have synthesized a nearly uncountable number of small molecule therapies. So on the right here, I have what, can be defined as a small molecule. And the thing is, when you're looking for new drugs to tackle biological problems, it can be very costly to actually test them. So we don't actually need to sort of make new drugs. This is more of a problem of testing all of the molecules that we have available to us, because you can imagine that the number of degrees of freedom that you have in chemical synthesis for something as small as this are more or less near infinite, right? Like you can imagine tweaking the angles on some of these bonds. You can imagine substituting these things for different chemicals, etc., etc. The amount of diversity in small molecules is unending, and in order to discover new drugs, it's more about testing those new drugs than it is about making them, if that makes sense. So we have a dearth in our ability to test said molecules. So how would you ramp up testing? So chat, I'm going to give you a little bit of time to come up with a product, you can sell it to me Shark Tank style, to come up with a product that would enable you to test a lot of small molecules for drug efficacy all at once, right? In the race against some of these terrible diseases, we do not have a shortage of candidates. We have a shortage of time and money. And this technology allows us to actually test a ton of different small molecules all at the same time, so that we don't waste decades trying to go over into testing things one by one. Microfluidic chips, interesting, Thamur. I would like to hear more. What sort of disease? Any disease you can think of. If you want an example, maybe cancer. That's typically a good one to go after. This drug test mouse is powered by AI. No, James. No, that's too real. That's too real. I do this to escape reality, not to be reminded of the reality that we live in. But yeah, so microfluidic chip, I would like to hear more about that. Could you just distribute it in water bottles? Well, not ethically, right? If we're talking about engineering, we want to make sure that we test these candidates in a very ethical way. And if you are distributing in water bottles and just testing it in people, that's not ideal, we'll say. Not ideal. Not ideal from the perspective of morality and ethics. But yeah, I want to put this question up because this is something that you could conceivably design. Broad spectrum antibiotics, that would be a type of molecule that we could try to test, but it's not necessarily an engineering approach. ELISA plates coated with target enzymes. Interesting. Say more gold. I think that's an interesting strategy. Plating on various media, is that not big enough scale? So media, you are implying that you are looking at antibiotics. So I'm talking about any disease, cancer, bacteria infection, viral infection. I think big with this. These small molecules could have potential to cure many, many, many different diseases. I thought you meant you wanted to put this out as wide as possible. Yeah, I mean more like testing these small molecules. So say you have 10,000 small molecules and you want to test them all for efficacy against cancer, specific kind. You can do pancreatic cancer, right? So you have 10,000 molecules. You want to test them all to see if it fixes pancreatic cancer. How would you do it? Inhibition zone tests. Yeah, that is specific for bacteria. I appreciate it, but I think our focus on bacterial infection might be inhibiting the ability to see the wider picture here. Certainly, I think the way that I framed it might have been falsely misleading people down the bacteria route. That's my bad. Think big. Think testing 10,000 molecules to cure cancer. I said antibodies, not antibiotics. Oh, gotcha. My bad, Vic. Also, let me reread your broad spectrum antibodies. Oh, okay. Yeah. So, so antibody is not a small molecule. An antibody is a protein. So we're testing 10,000 small molecules here. Certainly, like doing like an antibody pan is not a bad idea for a general strategy, but we're going from the belief that nature and chemical libraries exist, and we want to test all of them to see if there's potential there. So it's like a parallel approach. I'll say, make a bunch of mixtures and then do fancy statistics to separate what you want to test more carefully. Oh, so like a combinatorial approach is interesting, but I don't think that's super practical because you could have messy combination effects that would be difficult to untangle with statistics. Does this help the immune system or does it do stuff instead? So let's do like a very precise example. So let's say we want to find a small molecule that stops a cancer protein from functioning. I think that is the best way to put it. It binds, we want to find a small molecule that binds to a cancer protein and stops that cancer protein from functioning. I think I should have done that from the beginning, is provided you with that exact scenario. 10,000 small molecules, we want to find which of those bind to this cancer protein and stop it from functioning. Put them all in the same media and see if one of them worked, then use half of them to see whether that would work, and so on and so forth. That's an interesting approach. My argument is that with 10,000 candidates, that's going to take quite a bit of time and quite a bit of money. AI bioinformatics, that's an okay answer, but ultimately anything that you get out of AI still needs to be tested anyway, and AI is capable of false negatives, which would be ruling out things that could actually potentially work. So AI is definitely useful, but this is, if you want to be sure, you actually have to test it. So it is a good idea, a good application of it. I see the weary emote. But no, I think it's a good answer because it is an example where AI could be useful and not like a meme, right? Like a lot of AI applications these days are memes, and that is genuinely very useful, just not the answer in the engineering perspective, I'll say. Would you have exponential decay? I guess that's true. How many times would you have to have 10,000 for that to be effective? If anybody can do a quick back of the envelope check. So Gold, I assume you have biology background because ELISA is a biology thing, and we'll see whether or not your idea with the ELISA resembles the idea that is behind the small molecule microarray. An array of dishes would target proteins and apply a separate molecule to each one. Yeah, that's certainly an interesting approach. What's an ELISA? I'm not going to get into ELISA in this stream. It's a little complicated to explain, but it is reminiscent, I think, of what the answer is going to be. Just put the cancer cells in a test tube and a media, and then agitate, look at the cells, see what stayed connected. That could do it. But with 10,000 molecules, that means you need 10,000 wells of cancer cells, which is difficult to scale up. Create a GM bacteria/virus to create the molecules. The molecules we have no problem creating. The molecules can be synthesized. That's not an issue at all. Use robots and as many 96 well plates as possible to run many cancer enzyme inhibition experiments in parallel. Find which ones work, mutate the ones that do, and retest. Around 10 rounds would narrow it down to 10 molecules. I think that's interesting. However, you have to keep in mind that the first test, you're testing all 10,000 molecules, and then you're testing 5,000 molecules, and then you're testing 2,500 molecules. So that's still pretty expensive on your way down. So even though there are not that many rounds, the first rounds are going to be pretty pricey. Microarray plates. Okay, so we got some biologists here. Okay, great. So let us get into the brilliance that is the small molecule microarray. So the small molecule microarray was designed to capture what the inventor of this technology called pinches of magic dust. And I've actually been very fortunate to be able to witness a talk given by this person. And I think there's something so magical about calling this pinches of magic dust. So imagine you have a therapeutically relevant protein target, say the H protein on influenza, as shown by this arrow here, or the cancer protein, or whatever protein you want to drug against. The small molecule microarray allows for the simultaneous testing of millions of compounds against this protein all at once. And the question then is how? And so we've got some great ideas in the chat, a couple of which are hovering around the correct answer or are actually the correct answer. And so we'll see what we do from here. So the way that this technology works is you staple 1 million compounds to a slide. Here's my, a graphic representation of that. And so what you can do from here is you can then flow over your protein of interest. Say this is cancer protein or the influenza protein, whatever. Whatever this protein is, it's flowing across this plate with millions of compounds stuck to it. Then what happens here is that you wash off the proteins that didn't bind, right? This H protein is flowing over millions of different small molecules. It's not going to bind the majority of them, but it will stick to some of them. Then once those proteins stick to those small molecules, you can then use antibodies that are conjugated to fluorescent molecules that will allow you to detect the presence of a hit. And of course, because you built this microarray, you know exactly what these compounds are. So when you see a glowing light signal, you can actually find exactly which small molecules had bound to that protein. I think that is pretty incredible, right? Isn't that cool? I just want to sit here on this slide, if you've been following along, and kind of appreciate how incredible this is, because this is something that is not that crazy to think about, right? Like CRISPR is kind of complicated, right? It's kind of complicated. It's not the most complicated thing in biology, but all those diagrams, it can get kind of complicated. And with the golden rice, you have metabolic engineering, you have to know all these like chemicals and stuff. My prompt to you was pretty straightforward. My prompt to you was, I want to test 1 million different small molecule chemicals, and I want to test them against a potential protein target like a cancer protein, like influenza H protein. How am I going to do that? You build a chip, you chemically staple these chemicals on top, and then you just see what binds. And that is, I think, the heart of what I view to be brilliant engineering. Kajal says, is this the same process as ELISA plates? It's similar to ELISA plates. So ELISA is doing this, but instead of small molecules, it's proteins and in a well. So this is a microchip where you can stamp on these molecules. And ELISA takes place at a much larger scale. So we're talking about like, microscopic nanometer resolution on the chip is something that you can work with your hands, right? But like, these molecules are not visible, right? Whereas in ELISA, you do it in plates. So there's a lot more. ELISA is like a macro version of this kind of, sort of. There's also design features of the small molecule microarray that I'm not going to get into that help you figure out what the molecules are. But I'm, again, not going to get into that. But it is very simple. Doesn't the antibodies bind with the tip? The antibodies, you can have directional interference with regards to antibodies, but I didn't really draw the figure at all to be that representative, right? These antibodies are meant to just bind to the H protein that is left over. That's what I was wondering. How do they know that stapling it doesn't injure the molecule? That's an excellent question, Sierra Smith. So you there's some chemistry involved, and of course, if you find a hit, then you can always retest it, right? So this does not go into humans. This narrows down your list of 1 million to a list of 20. Then you can test that those 20 in more direct ways. But the idea is you can filter out a lot all at once very, very quickly. And I think that's pretty incredible. Doesn't the chemical stapling require a lot of per molecule work? That's why you build machines that can do it. You build a robot that does it for you. A very, very expensive robot for sure, but the chip itself is very simple. The engineering involved in stamping thousands of molecules onto a chip is a marvel in itself. Exactly. That is not easy to do. All right. Like, that is, the idea of this is easy. The actual implementation of this, of course, is a lot more complicated, but that's why this is a engineering 101 kind of thing. Are the small molecules randomly dispersed in the chips? If so, how do you know which ones get bonded to? So you know, because you designed the stamp, you know what molecule is where, because you have the blueprint of the stamp. I see what you mean. The idea of washing it off can only come from the mind of an engineer. I mean, some things are universal, I suppose. Okay, so I love this technology. This is like one of the things that I learned about while on the job, like teaching engineering from a basic science background. And I have to say that, like, to me, this is like the perfect piece of engineering. It solves a complicated, important problem. Like, not to say that your ideas were bad, chat, right? But this idea is like brilliant, honestly. And honestly, I could not have come up with this idea, right? If I were placed in your position, I would probably say something to the effect of microarray, but to have it be like this, I don't think I could conceive of this as even being a possibility. And so it's perfect engineering to me because it solves a complicated, important problem. How do you test all these small molecules that we don't have time to test? It's easy to explain, and number 3 is important because it makes you wish you thought of it first. It's handsome. It is handsome, right? Such a handsome idea. I think that number 3, if you have, like, I want to structure all of my streams based on this principle. Like, I want to make it so that you wish you thought of this, and that's how I want my streams to play out. And I think if I were to, I mean, the thing is that people who are in academia don't really need lessons from me, right? But on the off chance that there are people who become professors out there who want to do like a more entertaining job, I won't say good because good is subjective, but like if you want your slides to be more entertaining as content to keep your students engaged, setting up your lectures in which they feel like they could have come up with this, but then they didn't, that makes them more invested, because then it accomplishes a couple of things, right? It makes you think, oh, the person that made this isn't that smart. I could probably do it. And then it makes them think, well, why didn't I? right? And it's not because you're dumb, it's because engineering is fun and cool, and the easy solutions are often the ones that are hardest to come up with. So I think it's yeah, I just think it's so interesting. Okay, with that being said, I have only scratched the surface of biological engineering. In fact, I think in the pre-stream chat, I don't think I covered almost anything besides CRISPR that people were hypothesizing that I was going to cover. And that is because, of course, biological engineering is very, very deep. And again, just to recap sort of the themes of the stream, right? This field has deep roots in agriculture, but is very much its own thing, albeit it's a very young and rapidly developing field, right? And so what I hope we can come to appreciate is that leveraging biology to solve these issues is extremely, extremely powerful. But such power is not without significant ethical/societal impacts. So if you are like an up-and-coming biologist, if you want to get your hands dirty in this kind of field, I implore you, like, even if your peers are ignoring ethics and morality, don't be that guy, right? Don't be that guy that makes things for the sake of making things, right? Be responsible, be clear-headed, and talk to people in the humanities. All right, so that is it for my slides. I hope you all had a good time. I'm actually just going to end on this image of CRISPR while I go ahead and I take questions from the chat about biological engineering or biology in general for a few minutes before I call it a day. I certainly had a lot of fun teaching this in a casual format because, of course, you know, that like, you know, it's different when you teach students because they're expecting a grade, but just yapping to all of you, it's more casual and I think a lot more fun. Yeah. All right, so what do we got for questions here? 3 is super real. Yeah, exactly. Like the feeling that I could have thought of this is what got me into basic science also, because I'll talk about this tomorrow when I talk about my academic journey, but like the feeling of, oh, I could have thought about that. That's not so hard. Like that's an important moment, I think, for anybody who wants to do very, very complicated things, is the first time you feel that it is doable. You know, it's great because that's such an empowering feeling, because before you think it's doable, it's impossible, right? You get in your own way. So I will address Tan's question, but in a different way. So, you know, with CRISPR, it's inevitable that we come up with the problem of superhumans, right? Like this sci-fi idea that genetic engineering will create like a perfect breed of human. The problem with this is that genetic engineering is not riskless, right? CRISPR is actually not that good if you want to avoid accidentally getting cancer one day, right? CRISPR's success rate for only doing the thing you want it to do, not that high. And so there's a lot of safety issues with using it as a gene therapy. However, if you are going to die because your body is not functioning, then you can talk about risks of gene therapy, right? And so I am not personally that worried right now that that is an immediate issue, just because it is not feasibly safe to even attempt, right? The risks outweigh the benefits by so much. And we also don't have that deep of an understanding of the molecular nature of life to begin with. So even if you gave us the perfect tool today, making a perfect human is still not that easy. So I will say that it is something that we should all keep in mind as people like thinking about engineering responsibly. But it is not something that I would freak out about right now. But that's not to say you should forget about it. Just don't be afraid in the year of 2025, I would say. I think it's necessary to understand that easy solutions and solution easy to come up with are different. The latter tend not to be as good as the former. Exactly. And, you know, as much as I loved teaching my students, I will say that not all of their ideas were good, but it's very normal. Not all of my ideas are good. The process of having a lot of ideas and throwing ideas out is such an essential part of growing as not only just like, you know, an engineer or a scientist, but just like as a human. Try a bunch of stuff, and if you fail, it's fine. Working on various experiments is super fun. It is. Like, science is a lot of fun. Although I like teaching more. one of the reasons collaboration is so useful in engineering is because different people can come up with different parts of the idea and bring it all together. That's what's so fascinating to me, in my time as a teacher. Even though I'll be switching to YouTube full time, the time that I was an actual teacher, it was amazing to see what the students came up with, because they are all different from different backgrounds and different interests, ideas that I would have never have come up with. Some of them are closer to solving their problems than I will ever be. You didn't once talk about making xenomorphs by bioengineering? Certainly not for this stream. What do I think of de-extinction? I think it is flashy and not immediately all that useful. Preserving what we have, making sure we have sustainable ecosystems, probably a better use of our money, because the problems that caused extinction to begin with, just because you bring back something from extinction doesn't necessarily make our new environment hospitable to that species, right? So I think, as much as it pains me to say, but that whole direwolf business, I think, is hype, and I don't really care all that much. Although I will say it is impressive. Like, I do think that what they did to the direwolf, quote unquote direwolf, right? Not a real direwolf, but a wolf engineered to be similar to a direwolf in a couple of different ways. It's not trivial to do it, or it wasn't easy, but I don't really see the point. I'm not convinced, is what I will say. Well, I was here for barely 2 minutes. Will there be a video? Yeah, there's a VOD. There'll always be a VOD for the live. A video on it. Yes, so I will actually make a whole video where I go over biological engineering. I will talk about all the neat tools and tricks and all that good stuff. But that, I think, will be probably in like a year. Like, I have so much content to make. Like, I think, I think I die before I finish covering biology, which I've said multiple times. But yes, Wild Max, eventually I will make a full video on biological engineering, all the techniques and all the tools, because I think it's worth doing. And I think it's a good way to preserve my memory of engineering before I forget it, because I'm shifting to biology stuff. How much work is in the biological engineering in comparison to the actual cultivation with bioreactors and stuff? How much work? Like the job security and stuff? I mean, right now it's pretty hot, I would say, honestly. At what point does it become unethical not to use CRISPR? Exactly. That is not an easy question. And I think if someone has an easy answer, it's not well thought out, right? The answer to that is probably pages and pages and pages of philosophical discussion. If someone has a one-line answer to that, they didn't think hard enough. I like this video as an educational tool. I'm very glad you did. I don't think this is as much an educational tool for like teaching you how to engineer, but rather teaching you what is involved in engineering and teaching you like a little bit on how to think, or challenging you, because I didn't teach you anything in that regard, right? I didn't like show you how to come up with CRISPR. I just asked you and challenged you to think like an engineer, and then gave you the answer, which is how you are supposed to learn when you are taking tests, right? If you didn't know, if you want to get a better grade, take tests and then check the answer key. That's one of the best ways you can do to increase your score on any particular exam. Why is the strand of DNA like helix shape? Why not just a flat bit of thing like how proteins are? So proteins are also three-dimensional. We can even see one in my presentation here. Let's go back to going back a while. Where did I have? Oh, okay, well, I don't remember. Oh, here. You can see that the structure of the protein is not linear either, right? You have alpha helices, you have disordered regions or linkers, you have beta sheets, as you don't see in there. The point is that the way that amino acids link themselves up is not that dissimilar from DNA. Proteins can do more, but the idea is the same. The alpha helix, as you can see, these like barrels looking things, this is kind of like the DNA helix, so proteins are not flat little things. I don't know if our current level of ability to use CRISPR leaves any situations where it would be unethical to not use CRISPR. Yeah, I think that goes on the same path as where I was heading with my thought there. What would you do about endogamy afterwards? I'm not sure what that means. I think the direwolf thing was to get public interest and money to the field, not an end goal of its own. I think that's fair. I think it's very strategic that they wanted to build a lot of hype, but I think there's something a little icky about that, right? I'm a YouTuber, right? Like I get that you have to clickbait like a little bit, but I still don't fully understand. Like I read a bit about what they do. I'm still not entirely convinced that what they do is useful, even though what they do is impressive. I don't really know if it's useful. I've never heard of Talon until now. Will you talk about it? In my genetic engineering stream, biological engineering stream, for sure. Talon is very, very cool and important. I don't really know how it works right now, but it is CRISPR's precursor, which means that it's very definitely worth talking about. Will I do a stream talking about the older form of biological engineering, which I talked about at the beginning, like how we use selective breeding? So that's actually out of my expertise. So I'm not an ecologist, I'm not an agriculturalist or a botanist or anything, or even an evolutionary biologist, right? So those are a little outside of my expertise zone, I would say. I'm much more like a molecules guy. So I will have to be honest and say that I probably won't talk about that. Although that's something interesting for Nep to maybe tackle one day, is if she's interested in tackling the history of agriculture, which is not horticulture, right? It is different. But if she does have that expertise, that would be interesting to hear about from her. Yeah. And supposing you successfully bring back a species, how do you prevent it from going? Exactly, Seed, exactly. Like, I just don't find it very useful. What's your advice, pros, cons on becoming or not becoming an educator? Okay, so real talk, if you are in the US and you want to become an educator, you just have to kind of be okay with, you have to be mentally prepared to be poor for your entire life, right? You have to be happy with that possibility, because being an educator in the States sucks. You do not make money teaching. It has to be, you have to essentially realize that you are not happy doing anything else, which I don't want to romanticize, right? Like me, I'm extremely stubborn, where if I could make money by teaching and live quite simply, make enough to not starve, make enough hopefully to retire, I don't need vacations, right? Like, you have to figure out what you're willing to give up to be an educator, right? Like, are you okay not taking vacations, for example, like fancy vacations? Like, I mean, educators, you get summer vacation and stuff, but like, if you're in the US, are you okay with your vacation being, I go to my bordering state for a couple days? That's your vacation. You don't get to like take airplanes around the globe. And that being said, not all educators are like that. But if you want to entertain the possibility, if you want to live a life where you don't regret the choice, you have to be mentally prepared for that to be a possibility, especially if you teach K through 12. Now, if you happen to find that you are capable of being employed at a university or a fancy private school, then that's not as bad, right? But that's not a guarantee. I got very lucky in my position, is what I'll say. That is, of course, the con is that you just don't make money. But the pro is that it's extremely fulfilling, right? Like, getting to read my students' writing, like their first lab report, for example, where they don't know how to communicate a scientific idea, and then getting to watch their progress over the year. I get to watch their minds sharpen over time, right? I talked about this in my last stream, where you learn how to write. AI will never replace scientific thought, because learning how to write is so important. Because if you can't communicate your ideas in your own words, you actually don't really understand what it is you're talking about. And so you not only see their writing getting better over time, but also seeing their thought process clearing up. Because at the beginning, they can suggest an experiment, it makes no sense. Right? They're not thinking clearly because they can't. They don't have the words to think clearly. And if you find the idea of refining their thinking over the course of the year to be something that you could not be happy not doing, then yeah, become an educator. And of course, that's very specific to me, right? Because I teach engineering. But if if you're a history teacher, if you want to do history education, if the thought of getting, for example, students to realize the importance of civics over the course of the year, or something like that. If that is an idea that you think you can't do a different job, then then educator is great. Yeah, thank you for that ability, enabling me to soapbox one more time about education. For the practical limits of CRISPR, how much can you actually do with it? Can you edit? Like, I know there was a recent paper, I think, about the new size limit of CRISPR, but of course there is a size limit of what you can introduce. CRISPR is also kind of messy. It kind of inputs things where you kind of don't want there to be insertions, right? If you are trying to do a superhuman, CRISPR is actually pretty bad, because CRISPR is one of those things that could potentially lead to unintended mutations which could really hurt you. So that's why I think, it's part of the reason why superhumans is like pretty low on the list of things for me to be worried about. Endogamy is when related individuals breed with each other, reducing genetic variation. I don't have the expertise to actually know about what I would expect the ecological impact to be. So I cede that expertise there, seeding that to seed. Zinc finger as well in the MolBio engineering streams. Yeah. Eventually I'll be talking about the zinc finger. Only asking because the name is funny. It is pretty funny. And it did take like one class that I learned about zinc fingers, and it would be useful to refresh my memory because they're pretty neat too. Would CRISPR affect hereditary diseases? Only if you do the unethical thing of using CRISPR to engineer embryos or eggs or sperm. Yeah, no worries, Seed. Be nice to your teachers / professors, because exactly. And if you are somehow still taking classes, right? If you aren't like in summer vacation yet, if you're still filling out those teacher evaluations right now in the US, of course, if you have been paying attention, academia and the US kind of in a tense moment right now. So if you want to help out a lot, I'm not saying like suck up to every professor, but like if you enjoyed learning from a professor or a teacher, write them a really good review, right? They could use it in this very scary time. Being an educator in the US sounds difficult. It is, which is why a lot of educators often are enabled to be educators if they have partners that make a lot of money. It's very hard. It's not impossible, but it is very hard to live as an educator on one income. It just sucks, right? And I hope the situation changes where we can properly ensure that these, people who are responsible in large part for the way that the new generation will be trained are fairly compensated. Because like I'm mostly talking about education from like a college perspective where, the moment you turn 18, your emotional problems are no longer the problem of your educators, which is kind of heartless to say, but it's kind of true. Whereas like K through 12, when you're 18, your teachers play a huge role in your mental health, your maturation as a human. You get to university and the responsibility is, professors can certainly be a part of the lives of young adults, but at the university level, then you can have access to therapy that is compensated by your university. And so a lot of that gets offloaded, at least for private universities. If you're a public university, I don't actually know if they cover therapy and stuff like that. But the idea is that like K through 12 is so hard. Educators are not just teaching, they're also helping children become fully grown adults, and they are not nearly paid enough. A little off topic, but I often hear that people go into great debt wanting to study when it comes to studying sciences in the US. If they are already doing so much, where's the money going? Yeah, so if you want to get an undergraduate education, you have to pay for that. If you qualify for financial aid, certainly do apply. You can get a lot of that money compensated, which is good. If you can apply for state universities, if your state university is quite good, then go to that. That saves you a lot of money. Community college saves you a lot of money, and then you can get scholarships, which also saves you a lot of money. There's an issue with compensation in the US where tuition costs have bloated, but the supplemental income from investment from the government has remained pretty much the same. And so education costs keep on increasing, and there's nothing that is really done about making sure that people can still afford that level of education. And like, I can't claim to know exactly what happens to tuition money in like super detail, but like paying salaries, of course, universities are in essence like centers of towns, right? You are essentially funding an ecosystem. Now, do I think that tuition costs have ballooned very high and too high? Yeah, like tuition is kind of crazy. And so like getting financial aid is therefore even more important, and having the opportunity for the government to invest in its students, that is, I think, in the interest of every government. That makes sense, right? You want your students to be highly educated and able to produce value in the capitalist system that exists in. So of course I think that more investment into giving students a better education for the production and health of a country is very, very important. But yeah, it's tough in the US because conflicting interests makes this very, very difficult. And also, like, going into debt, having to pay on the loan is very profitable for companies that give out loans, which is very messy. And I don't want to get into that so much as to say that at the end of all of that, if you choose to go for a graduate degree, like a PhD, you can often earn money on the side and have your tuition paid for. So people do go into debt to study. If you go into advanced study, it's not quite as bad. But if you can get a job right after undergrad and make a lot of money that way, that's probably your preferred path. Probably your preferred path. You had ethical concerns about teaching us the how-to. Should we be afraid of some guy asking an AI how it works and doing something stupid? I think stupid people are always something to be afraid of, right? And there's only so much that you can do to prevent people from getting access to tools, but because of the internet, that is always something that is a possibility. However, I will say without the proper training, good luck. And without the proper material, good luck. Right? I said CRISPR can be done quite cheap. You still need a lab to do it. You don't do it for free, right? It's still expensive. It's just of the gene editing tools, it is of the cheaper tools that you could do. It's again, it's still not free. I'm at Purdue, and at least here there's a university counseling service, and it's a public land grant school. That's really great to hear, right? That's awesome. Yeah, but that does also mean that your emotional connection, your mentorship relationship with your professors can be quite varied. Now, some professors are quite happy to mentor students, not all of them, because they are, of course, very busy. But like most of what the professor is doing is teaching, writing grants, training grad students, not so much walking you through how to be a functional adult. How is it unethical to use CRISPR on embryos and sperm if it is to take out a disease that's running in the family, like Huntington's? It's unethical because where do you draw the line, right? Say, for example, somebody wanted all of their babies to be super strong and super smart. Well, you know, hypothetically, again, it's too complicated. It wouldn't actually happen because we don't really understand biology to this depth. But imagine that there's super smart gene, right? One gene controls super smartness. You could potentially insert that into an embryo and then have everyone's child be super smart. Now, there's an issue there because who gets to actually do this? Well, of course, the rich. And if you give the rich an advantage, that is just one more way in which the rich can suppress the poor, which we do not want to do, right? Engineering should be thought of as a way to make people's lives better, not to increase the gap by which the people whose lives are already pretty good get even better from there. So that is the way that I would explain why using CRISPR on embryos is an issue. It's because those are permanent and inherited. So you can create like a subsect of people who have even more advantages than we're comfortable with having right now. Yeah. How much can you alter before stepping into dubious territory? I think you stay away from the germline 100% of the time. You do not touch sperm, you do not touch eggs, you do not touch embryos with CRISPR. I think if you have a bone disease, you can touch bone. You can touch stem cells that become bone. I think that's cool. If you have a skin problem, you can use CRISPR on the stem cells that become skin. You do not touch sperm, you do not touch egg, you do not touch embryo. That's where I would draw the line. I would be happy to be convinced otherwise, but if you want to know my current stance, again, science is all about updating your beliefs over time when you learn new information. Right now, with the information I have, no germline stuff. All right. That being said, I will now call it a stream. Had a lot of fun here, y'all. Yeah. Oh, thank you, Toxo, for the end of stream plug to support Fi if you are financially able to. Thank you so much, Toxo, for that plug. I got a little too lost in the sauce. Of course, I am going full time now, away from my job as an educator at a university and committing full time to making YouTube content. This week of streams is to kind of shout out that membership, which I did a really bad job doing. But if you click ! membership, you will be reminded that that is the case. If you try to join membership, you can actually see the different tiers that you can apply for. I'm pretty sure it's been updated. I didn't do like a walkthrough of the membership today just because I wanted to keep the VOD pretty crispy, but also it takes a little bit of time for the YouTube membership to actually update. I think it should be recent now. At the T-Cell Award reward tier, that's where you get most of the benefits, and then above that is like, you know, you just want to give me money to support what it is that I'm doing here, which I'm very happy to take. But I think for most people, if you want extra content from me for $5 a month, you can get that extra content and be involved in some gaming streams that I'll be doing. So I'll be doing like monthly gaming streams that you can be involved with. Of course, the higher tier you subscribe to, the higher priority you will have for queuing in those games. It's just a way for me to make more content for you and to get you involved in the community. I think would be really fun. All right, with that being said, yeah, thank you all for coming to my talk about biological engineering. I hope you've had a great time. Tune in tomorrow for a more in-depth walkthrough of what my membership now means, as well as talking about a lot more about my academic journey from when I was very young to where I am now. And I think it's interesting to kind of reflect on that journey because, of course, the opposite side of teaching is learning, and to talk to you about what that was like for me, I think is going to be pretty fun. Yeah. Thank you all for coming. I will see some of y'all, hopefully most of y'all, tomorrow. Bye-bye, y'all. Bye-bye. Bye-bye.